good morning, folks. I'm Eric Burris. Thank you for joining the California Water Quality Monitoring Collaboration Network. We have a very special webinar presentation to you today on ICON. It's a really neat collaboration, potential for everybody here in California. There's probably a site near you. I will let our presenters cover all the wonderful things that the project is doing and how we can participate. This webinar is being recorded. So coming to us today is Dr. James Stajan. He's an earth scientist with the Pacific Northwest National Lab. He's an international recognized expert in ecological theory, the interface between microbial ecology and hydro-biochemistry. And he's looked at distributions of open watershed science and is a leader in that. Also with us is Dr. Stephan Gary. He's a senior application engineer and computational scientist, Parallel Works, is a physical oceanographer with 15 years experience observing and modeling the North Atlantic Ocean. So thank you, gentlemen, for joining us and giving this wonderful presentation and also collaboration opportunity.
Excellent, thanks. Before we get going, so Bree and then Lupita, can you introduce yourselves? And then Lupita, can you talk about PNNL?
Hello, my name is Brianne Forbes. I'm a postmasters at PNNL. I work on data management and publishing our data, open source data, publicly available, and I also work on a lot of the logistics for this project.
Can you hear me now? Hello, I'm Lupita Renteria. I'm an earth scientist and I work on river corridor research with James and Bree and Stefan. And I do a lot of some of the field work and a lot of the lab work for this project. And so now a few things about the lab, or I call it the lab, but formerly known as Pacific Northwest National Lab, PNNL for short. And we have a main campus here in Richland, Washington, but there are other locations in like Sequim and DC. We're made up of about 5,000 employees, and we're funded by the Department of Energy, Office of Science, but we work for a private nonprofit called Battelle. And so we don't directly work for the government, but we are contractors. And we're a research facility that does a very wide range of research, from environmental to nuclear to human health, and it varies from like applied to fundamental. And we have lots of opportunities to have others come engage with us. And so if you or anybody else is interested, please reach out to us. And yeah.
Nice, thanks. I may have missed it if you said this. There's like 5,000 employees, like pretty big place, and internships from every single level, from undergrad all the way through senior scientists. Stefan, do you want to just briefly describe Parallel Works, and then I'll launch into the slides?
Sure. Parallel Works is much smaller than PNNL. We're a 15-person startup originally based in Chicago, but now fully remote. And we make it really easy for folks to run high-performance computing in the cloud. And so in the partnership here, I'll be talking about running the machine learning on cloud resources. So thank you.
Okay. Thanks for getting us going. Thanks for the introduction, Eric. And so in terms of rolling on here, what we're going to talk about today is a project that we on our team refer to it as the ICON-ModEx project. I'll explain what that all means. And this presentation, first of all, I think I put it in the chat. Yeah, it's linked in the chat if you wanna have a look for yourself. And we're gonna go over a few things. We're gonna go over ICON science. What does that mean? And we're gonna go over then the application of that approach in the context of river corridor science. And talk about some science motivations. We'll talk about our technical approach, and we're going to talk a lot about opportunities for folks to get engaged. And this is really anywhere from citizen scientists all the way through professional researchers, students, their opportunities across the board. So let's dive in then. We're going to start with ICON Science. What is that? Well, it's an acronym, and it stands for the 4 words here: integrated, coordinated, open, and networked. And really what ICON is it's a framework or a heuristic through which we can use it to help the intentional design of any project that aims to generate outcomes that have mutual benefit between, say, a project team and stakeholders that may be beyond that project team. How do we actually have true mutual benefit in that way? And to simultaneously generate knowledge that is transferable across systems. The science that we do, we've moved away from case studies per se and are generating knowledge that is transferable and generalizable. And so how do you actually achieve those things, mutual benefit and transferability? And ICON, as I was saying, is a heuristic, a framework to help us do this. And it applies across all science domains. We're going to talk about it today in the context of rivers, but it's not limited to that. But what do we really mean by these 4 words? So ICON Science is intentionally integrated in the sense that as we design projects and as we implement them, we're very conscious about how do we link together physical, chemical, biological, and or social attributes, and how do we do that across scales? That may sound simple in some ways, but we have found that that takes care and attention to actually do that well. How do we purposefully, in terms of being coordinated, how do we purposefully use consistent protocols and consistent methods through space, through time, across research groups to enable transferability, interoperability? If we all use different methods, we end up with a splintered collection of pieces of data and knowledge that are pretty hard and almost impossible to put back together. So that, again, requires care at the upfront side. Open throughout the research lifecycle, sharing ideas, sharing data, sharing protocols. That's really quite vital in order to be able to generate fair data that is findable, accessible, interoperable, and reusable. Current paradigm in many scientific domains is collect some data, publish the paper, and then throw some data on the web, and okay, you made your data available, but it's not really that usable. And being open throughout the whole research life cycle is quite important. And then lastly, ICON is about being intentionally networked in the sense that a research team can work purposefully to understand the needs, the values of stakeholders beyond their research team. You know, a given research team is funded to do a particular thing, but oftentimes there's little tweaks that can be made along the way that increase the value of that research to those beyond the research team. Well, how do you know what to tweak? You need to engage people and learn and listen, and then make sincere shifts in what you're doing. And that's where the mutual benefit comes from. Doing science that is true to what teams are funded to do, but also providing outcomes that are useful to others. And what we have found is that most people can relate to these concepts, and a lot of people think that they are doing all of those things. What we have found is that it actually takes, as I was saying, a lot of care and thought at the front end to truly do these properly and well. And so we've launched a thing that we call the ICON Science Cooperative to help with this, to collate experiences, knowledge, methods, know-how, how you actually do science in this way. And it's, again, meant to be across all disciplines. So if any of that is of interest to you, I don't know everyone's backgrounds or interests or whatnot. If any of these concepts ring true to you, please reach out to us. There's an email address there. There's a website. You can check it out. We'd love to engage with you. And there's lots of opportunities to do that. We can work with people if they're writing proposals. They want to put ICON in there. We've done that, where we worked with a project team. We put ICON in their proposal, and they got their proposal funded, and the reviewers were very complimentary about the inclusion of ICON. So it can add practical value in terms of getting proposals funded. We can help with existing projects to bring more ICON in, hands-on implementation. We can host students. So Lupita was talking about this in terms of opportunities for people at PNNL. We can host people interested in trying to do ICON in their work across all levels of engagement. There's some other opportunities there. There's, again, an email address. And then what we'd like to do, you can take a moment, and if you have a phone, you can scan that QR code that's on the screen right now. What it will do is we'll take you to a Menti survey. Very simple. It'll take you about 15 seconds to fill that thing out. Just to give us an idea of like, does this make sense? Icon, does it make sense? It's also a moment we can just kind of pause and see if people have questions or comments or suggestions about this framework. Again, of thinking about it broadly. We're going to dive into rivers in a moment, but thinking about it broadly. Would love to have your questions. If something's not clear, if you have an idea, or how can, this seems connected to this other idea. So I'll just pause for a little bit, and if someone has a question, throw your hand up or put something in the chat or just unmute. Okay, so no questions, all right. So if you can, it'd be really helpful for us if you can scan that QR code and take that little survey. And I think Bree will grab the results of that and throw it in the chat if she has time. So, okay, Icon, let's turn the corner into rivers. So example of Icon in practice, river, and this is this river corridor project that we call ICON-ModEx. So what's kind of our very simple high level motivation? Rivers are obviously really important to the whole Earth system. They're quite vital in terms of the biogeochemistry, the elemental cycling of the entire Earth system. They're of course very important for local issues, water quality, water availability. And something in that context that we're motivated by is the observation that within rivers, if you think about the sediments, if you're standing on a river, you got sediments under your feet, that zone is very biogeochemically active. It's often known as the Hyporheic zone, basically where surface water, the river water, runs through the sediments and comes back into the channel, this kind of mixing zone. Very biogeochemically active. However, it contributes anywhere from 4% to 96, and we've even seen 99% of total integrated stream respiration and stream metabolism. And that metabolism is a vital part of the river system, right? That's influencing how much carbon dioxide and methane and nitrous oxide are going to the atmosphere versus how much is retained in the system. Strong influences over contaminant transformations such as nitrate. So it's this vital integrated measure. These sediments are active, but they don't necessarily contribute a lot. But sometimes they do. But there's no model to help us predict that or understand that. So we're inspired by that kind of that challenge. How do we actually gain traction and be able to predict variability in hyporheic zone or sediment biogeochemical contributions and the rates within those systems? Okay, so how do we do that? We're going to use this consortium that we lead. It's called WONDERS. I'm not going to go over the acronym. It's kind of long. The point of WONDERS is it's a consortium that's enabled by the science community. WONDERS is focused on studying river corridor hydrology, biogeochemistry, microbiology, doing interdisciplinary science, and producing outcomes that are useful to the community. Again, like from an ICON perspective, this is sort of for and by the community, basically. And I'll give a little bit more details of how WONDERS really operates in a little bit. But that's a key kind of vehicle for actually doing the science. Okay, then we're going to start to dive a little bit more into the science itself. So what we're doing is WONDERS is based off of crowdsourcing samples. So in 2019, we crowdsourced samples of riverbed sediments or hyporheic zone sediments from across the US. And those came back to us at PNNL, and we measured respiration rates, like the consumption of oxygen on those sediments. We did that in the lab. So now we have respiration measurements made using a consistent method across the nation. Again, that coordinated perspective from ICON. And then we made the data open, publicly available, machine readable. It's fair data. And okay, so again, ICON's coming in here. So then what was cool is then Stefan was like, ooh, there's data there that can be used to start to build, test artificial intelligence methods, cloud computing methods, and do some science and try to address this particular science challenge that I was mentioning on the last slide. So he took those data and he built a suite, a collection of machine learning models that are then predicting respiration rates at thousands of locations across the nation. We sampled, the consortium sampled about 90 locations. And then he's taking those data, building machine learning models that now predict across thousands of locations. So from our perspective, well, that's cool. Now we have predictions, which to us means a hypothesis. We have a model-generated hypothesis. What should respiration rates be at all these locations? So now what we're doing is we're going from machine learning of making predictions into artificial intelligence, where now we're trying to use those models to help us make decisions about where we should go sample next, to test the models, improve them, and generate new understanding and predictive capacity. So this is, again, where crowdsourcing comes in. What we're doing right now, the current project, is Wonders is, again, the vehicle to work with the community to crowdsource samples at specific locations across the US. And those locations are guided by the artificial intelligence. That's the decision-making piece here. So we send out sampling kits. I'll show you those, I think, on the next slide. They go to people, standardized protocol, everything's self-contained. We pay for all the materials, all the shipping. People need very little of their own materials and resources to do this. It's really about expanding people power. And kits go out, they come back to us, we generate the data, we publish it, we make it available, and then Stefan takes those data. Bree handles a lot of that. Lupita's engaged a lot with the generating of the actual data. Bree sends Stefan a file, and then he takes those data, tests the models, and then rebuilds the whole system. This happens monthly. And so we're going around this cycle that we call ModEx, Model Experiment Iteration. Every month we go through a ModEx cycle and we get new guidance. Where should we go next? Where should we go next? And we reach out to people. Bree does almost all the heavy lifting of reaching out to people, engaging with folks like, are you interested? What could be useful to you? Are there places nearby that you could get to? These sorts of things. And thank you, Bree. She just put a map of the current map of the locations that are of interest. And by of interest, that means the artificial intelligence is sort of telling us to go to those places. So then PNNL, as I was saying, we send these kits out. This is the Wonders piece here. So we build standardized kits. Lupita does a lot of that hands-on work. And they go out to folks. Sampling is done. We have very detailed written protocol, a video protocol, a video about how to avoid contamination, trying to be as consistent as humanly possible. And so the kits go out, sampling happens, takes a couple hours, they ship back to us, we receive, we process, and we publish the open data. So here's boxes, coolers, teams processing samples. There's vials to get water samples. There's bottles for sediment samples. There's vials for microbial analyses. We have consistent metadata that are collected in the field. That's in the bottom left kind of image there. And to make decisions about what we were actually going to collect in terms of sample types, what analyses we're going to do on those samples, what metadata are we going to collect, we held open community discussions and we asked people, like, what's valuable? Here's our ideas, but they're just starting ideas. What's valuable? What's useful? And we incorporated that knowledge, that information from the community, vastly expanding the brain power beyond our individual team while understanding what other people value. That's the networked part of ICON. That's really important to us. We made a number of tangible changes to what we were doing. And we're getting a lot of diverse samples back in this process. You can see a picture here, just the different colors of sediments from across the nation, which I just love that picture because it's just like, yes, we are doing science that can be transferable across diverse systems. That's really vital. Anyway, some other photos of processing samples. So it's this is what I mean by crowdsourcing. Let's see. So with that, I'm going to hand off to Stefan. I'll drive slides for you as needed. And take it away, please.
Great. Thank you, James. At this point, I thought it might be useful to try to dispel a little bit of the mystery that surrounds the machine learning and really say, sort of say, this is what we're actually doing. And because machine learning is often used as a catchphrase, and it's really nice to drill down into specifics. So here's an example of what we're doing. The data that we're using is organized in a table where the columns are going to be the inputs to the machine learning model. So that would be on the left box. And the one thing that we're trying to predict is, as James said, that's the respiration rate in the sediment. And so that's milligrams of O2 per liter per hour. And so each row in this data table is a site, and we'll give it an ID at each site. And so we're using data at these sites to predict the respiration rate at the site. And I'm using way more features or inputs than are shown here. There's right now the models are using like 25 different inputs to predict what the respiration rate would be at a given site. And when the new data is available, Bree sends me a file. There's about 400 something sites now, and there's a respiration rate. And that data set is used to train the machine learning models. And then basically the goal is to say, train the model so that for a given set of inputs, make a prediction of respiration rate. Once the model is trained, I can just hand it a bunch of inputs, these features, so temperature, pH, oxygen, for example, and then it will make a prediction of the respiration rate at a site that we haven't sampled yet. James, could you move to the next slide, please? So here's another example, or sort of a top-level overview or schematic of what we're doing. At the very top are a list of 3 possible sites. We have a latitude and longitude, so where they're located. The collaborators have collected information about the water temperature, the oxygen, and the pH. And I can put that in the machine learning model, and it'll spit out a prediction of the respiration rate. So that column in the box, in the bottom left box, where those are at BCR corresponds to the Buck Creek Road site in the top table. So for each of these sites, we get a prediction of the respiration rate given these inputs. The other thing that I'm doing here is that bottom right box, where in addition to these local variables or local inputs, I'm co-locating this data with large-scale databases. So when James mentioned integration, or the I in ICON, like the integration across scales, looking at data that's at that location, but also looking at larger scale data, this is the integration. Part of the integration is I'm also using the latitude and longitudes at each site to look up information at that site associated with river flow, climate indices, land use indices that are in basically global scale data sets, but taking that information and putting it at that location. And so those are also used as features or as inputs to make the machine learning predictions at the sites. The other interesting thing that's happening is we're making predictions, as James mentioned, at about several thousand sites across the continental US. And those sites are also the information that we're using to make these predictions are pulled from these large-scale databases as well. So basically, anywhere I can find latitude, longitude, water temperature, oxygen, and pH, I can then attempt to make a prediction at that site. And the bottom boxes show the global averages of what the respiration rate is across all the sites I have access to and their standard deviation. And you get a rough idea that some sites might be higher or lower respiration than that average. And that's useful for helping infer which sites we want to sample at. We really, to kind of guide this, what James mentioned is artificial intelligence, which is deciding which site or putting a metric to measure which site may be of most interest. We combine 2 things. One of which is we combine the error that the machine learning model thinks it has attributed to each prediction. And the second thing we add is we compare all the sites relative to each other, and we say which sites are the most different from the others. And by combining those 2 metrics, the error estimate that we have and how different a site is from the others, we then produce this metric column, which is that's alongside the prediction for respiration rate. We have this metric, and that metric tells us whether that we rank the sites based on those metrics to see which may be of the most interest for sampling. The plot on the far left side is an example, a scatter plot of this metric, basically shown in 2 dimensions. So there's a question here. To what extent do you manually verify the AI predictions for quality control? This is great. So in what we do is the information of like the ranking of sites of interest, that's sent to Bree, who then puts it in the map that is in the chat. And so part of that manual verification is looking at which sites are of most interest for a given, it's not, we're not going from machine learning to automatically writing an email to a collaborator saying that site is of interest, but there's definitely a manual inspection that says, oh, these sites may be of more or less interest, but we're going to, verify that first. Was that helpful in answering your question? I'll pause for a minute, and thank you, James, for moving to the next slide. Great, thank you. This slide here is showing kind of what our plans are for future work. The slide on the section on the left shows the score of the model, or basically the skill of the model, how it evolves as we go through the number of ModEx iterations. We've now done 9 complete cycles, so this slide is slightly out of date, and that percent variability explained is rising as we add more and more data, as it should. And for future work, what we're interested in doing is applying some machine learning techniques to identify which of the features are the most important. We're currently using 25, but we could use more or less. We're applying a new technique to boost the training data with synthetic data, and I can describe that later. And we're also interested in basically plugging in this model into larger models like Earth system models, so that those models can get estimates or predictions of respiration rate within that larger model. And finally, the piece that that's happening most immediately right now is that our goal is, by the end of this month, is to automate, well, in a few days when Bree sends me the next data set, is to fully automate end-to-end this process where the new data is available and it kicks off the training, which happens in the cloud, and the models and the predictions are automatically pushed back into a public GitHub repository. And I can go into more detail on that later. James, next slide, please. So here's an example of how going from one cycle, one ModEx cycle from here's an example of when we did our first predictions. We had we plotted the sites of most interest, the top 10% sites that had the highest metric, and then when we added more data, trained a new model, those top 10% sites shifted in space because their metrics changed relative to each other. The predicted respiration at the site and the error on that prediction changed with the new model. And so this shows that over time, as we add more data, the models are making different predictions, but the cool thing is that it's starting to stabilize. And so these dots aren't moving around as much. James, next slide, please. Thank you. And here is an example of the map that we share with collaborators that shows the sites that have the highest metric, which are in blue, or I'm sorry, those are in the purple stars, and then the sites that have a lower metric, so they are of still interest because they are useful for verification of the model. And let's see, next slide. Oh, perfect. All right, James, go for it.
Yeah, cool. Thank you, Stefan. So One thing with machine learning and artificial intelligence is it can provide interesting predictions, it can provide hypotheses, but in some cases it's a little bit black box and it's not necessarily giving us mechanistic understanding. So there's a difference between being able to mathematically predict something and actually understand the governing mechanisms. Those are two different things, related but different. So along with generating the respiration rates, which we, as I've mentioned before, we do in the lab, we're also generating a variety of other data types. And these are much slower to generate. So they're not really conducive to this month by month iteration. But again, these are data types that we gathered feedback from the community in those open calls I mentioned previously. And we put questions to the community. Would this be valuable? Would this be valuable? What do you think? And this is the list that was kind of zoomed in on, I suppose, based off of feedback. And so for water samples, what we're getting is organic carbon concentration, total nitrogen, major anions and cations. We do very high resolution mass spectrometry. That's the FTICR. That characterizes the organic molecules that are in the system. Gives us very deep information on organic chemistry, oxygen, temperature from the surface water, from the sediments. We again get that very high resolution organic matter characterization. Same thing with the ions, major cations and anions. We're also pursuing a different kind of mass spectrometry known as liquid chromatography that is really good at of quantifying, observing small molecules, so more of like microbial metabolites. We're doing the respiration incubation, so as I mentioned a number of times. We're doing mineralogy of the sediments, the carbon and nitrogen on those sediments, the grain size distribution, the moisture content when we receive the samples. ATP is a proxy of active microbial biomass. We're sequencing the microbial communities, metagenomes, metatranscriptomes. We get photos that we can use for grain size analysis at kind of the larger scale. There's some other bullets at the bottom. I'm going to skip those for today. The point is that as time goes on, we will be generating a very, very rich suite of data that span physical characteristics, chemical, biological, in order to put that together for integrated understanding, mechanistic integrated understanding. We'll also feed those data through the and ask the question, if we use those data in the machine learning, what does that do to the model performance? I'm going to pop back here just for a moment. If you notice the values here on the vertical axis, percent variability explained, at least at ModEx level iteration 5, we were only hitting about 25%. So what that says is most of the variations actually not being explained or captured by the machine learning. So I'll just flip back. So one hypothesis is that these kinds of data are not, or most of them anyways, are not currently used in the machine learning models. Well, we might be missing really important detailed molecular processes and features and properties that we will have data for. And we can then ask the question, well, does the machine learning performance increase dramatically when we include all these detailed molecular properties? My hypothesis is that yes, it will, because I think what's going on is there's very localized control from site to site, from river to river. Very kind of nuanced, context-dependent things are happening, and it boils down into the molecular properties. But time will tell, and all those data will be openly available as well. So this is our last slide. So we should have a good chunk of time for discussion. So get your questions ready. And so let's talk about opportunities. So we talked about icon science. We've talked about the technical aspects of this project. As you have seen now, this is this crowdsourced effort. The maps are changing every month of where the models are suggesting we should go. Bree is working hard to schedule people every month to collect samples. And so, but there's really a whole menu of ways to engage. So we're doing virtual presentations. This is one. We're going into classrooms to do these kinds of presentations. At the moment, we're doing that at the university level. Bree has connected us with a group that we're going to start developing some materials for a younger age cohort, more in the like middle school to high school kind of range. We're excited to do that to broaden our engagement. For those of you that are faculty, or not necessarily faculty, but research kind of folks of various flavors, we could partner on icon-based proposals for whatever science domain you may be working in. River corridor specific proposals, that could be icon, that could be our technical aspects, what we do with river corridors, crowdsourcing, molecular analyses, whatever it might be. Computing proposals, more with Stefan and parallel works and cloud compute and AI and machine learning. Those are of great interest as well. As we've already mentioned a couple of times, there's a lot of different kinds of student opportunities. Students, and really anyone, can use the data, the models, it's all open. People can use the data to go and write their own manuscripts. We're also going to be launching a effort fairly soon in which we're going to start writing a manuscript around for this project, and we're going to do that in an open way that whoever would like to can contribute ideas, analyses, drafting text, finding literature, whatever it might be. People can engage and actually being part of that manuscript process. Lots of internship opportunities at every level, even all the way down to high school, can come to PNNL. We have a pretty big collaborative network. We can help open career opportunities for folks, kind of opening doors, expanding collegial networks. We can train people. We're generating a bunch of different kinds of data that a lot of people may not know how to use. We can train people on how to use those. Just as an example of one flavor of training. If people are interested in collecting samples, that maybe there's a river that you care a lot about and you want to know more about, you want to get some data, well, please reach out. We can send you a kit and get things going that way. So you can get data for your site of interest. And that's basically, if you do that, you're inherently engaging in this ModEx process. You're part of Wonders, you're part of this project. We very much view this as a big collaborative network. And more than just collecting like a whole kit of samples, you can also just go to any stream you like. Does not need to be a stream that's a priority site. And you can just give us pH, temperature, and oxygen. That's it. And the coordinates just from your phone. Send them to us, and Stefan will run those through the artificial intelligence, and it will tell us, hey, that's a priority site, or maybe not. It might fall in the middle and it may not be a priority site, but that is available to any stream anybody wants to go to anywhere in the nation. And if people don't have an oxygen sensor, sometimes that's a limitation. We can ship oxygen sensors around, so we can facilitate that way. And so there's another QR code there. If any of that is of potential interest to anyone, I would encourage you to scan that QR code with your phone and just let us know, like, hey, yeah, interested in sampling, or interested in manuscript help, or interested in the data, or interested in training, or collegial opportunities, or whatever it might be. We have a variety of menu of options. That's a Google form. That's a different QR code than you scanned earlier. So with that, that's our slide deck. I'm going to turn the slide share off right now so I can see people. It's very easy just to start it up again if anyone wants me to go to a particular slide. So with that, would be very happy to answer questions, get suggestions. Often when we do this, people actually ask us questions that are pretty penetrating and give us new ideas. We're always interested in that feedback and trying to do things better. So yeah, I'll stop talking and let's have some discussion. Thank you.
Thank you, James.
Okay, so we already have a question in the chat. This might be for Stefan. How does flow rate impact your predictions? Or does it?
I don't know I've not looked at that one directly. That's a great question though. And I can verify that. One of the classical way to test that is a technique called feature permutation importance, where you look at the predictions that you make using your data set, and then you take flow rate and you scramble it across the whole data set, but keeping all the other variables the same. And if your predictions get completely, messed up and get, are no longer any good, then you know that it's super important. But that's indeed something that I'm planning on doing in the next stage, because in the past we've done this kind of analysis, but we didn't have enough data to really have a lot of confidence in it. But now that we have a lot more data, it's a good time to answer that question. So thank you.
And one thing I'll just continue on there a little bit of a story, I suppose. In doing this kind of outreach and talking to people, there was a researcher that reached out, an early career person, and he's a hydrologist, not really into the the organic chemistry necessarily or that kind of molecular stuff, but a hydrologist. And he filled out the Google form. He's like, I really would like help with kind of connecting into science efforts and like getting engaged. Help me with my scientific collaborative network. How can I partner with you in like writing manuscripts and on that side? And I mean, he's going to go take a sample, but that's not really his main thing. He wants to engage in the science and bring in the hydrology, which the kind of the core team here, we're not really hydrologists. And so that's really cool. I mean, we have hydrologists, around at PNNL for sure. But I think this is neat, where what we've been doing is engaging and trying to figure out the right, like, what is he interested in? How can that benefit this project? How can that then benefit him in a career perspective? And so as we open up this manuscript development opportunity that I was mentioning, he's going to engage with that and bring in that hydrology perspective, and I think really help us press on those questions, such as how does flow rate impact your prediction? What does it really mean? How do we access, what are the right kinds of data to actually be using to even test that? Most of these sites are not gauged. There's no like stream gauge at these places. So, okay, how do we actually bring flow rate in or discharge? So I think that's going to enrich our science and help him at the same time. I just love those like intersection points to achieve mutual benefit. And it looks like there is a hand up, Patrick, maybe?
Yes. Thank you. I asked those two questions. First, my name is Patrick Mbaba. I'm a petroleum engineer with the Bureau of Land Management, Department of the Interior. What you're doing is very important, very critical. However, I see a platform of, similar to reservoir simulation, because you have some movement of the data that you're measuring. I think some application on reservoir simulation application could really be helpful to authenticate the predictions that you have, because it's going to change almost every day. So, but I want to congratulate you for the data structure that you present. At the same time, you may want to consider that feature to improve accuracy in the in your predictions. That's the comment I had. I asked the two questions about the manual verification and also the flow rate, because of the fact that you're dealing with hydrology there. Thank you.
Yeah, appreciate that feedback. Bree, hopefully this doesn't catch you off guard at the moment. Do you want to speak to what we're doing with Neon for time series and the gauges they have and how that might relate to what Patrick is saying?
Yeah, so NEON is the National Ecological Observatory Network, and we have 3 spots across the U.S. Where they're taking repeated sampling once every other month. So we'll get more of that temporal aspect from those 3 sites.
Yeah, and a cool thing about one of the nice things about Neon is those sites have stream gauges, and so we can see the hydrograph and what's going on and flow variability and what was the flow when we took those samples, and how does that differ from time point to time point, which isn't quite, reservoir operations, but at least I think it starts to head hopefully down that direction, Patrick, that you're referring to, of temporal variability and how the surface water hydrology may influence those dynamics. And we'll see. We don't really know yet. That I think NEON has taken maybe 2 samples thus far. And those are, at the moment, those are the only sites that we are repeatedly sampling through time. So I think that'll be really important and interesting to see what comes out of that and how variable things are through time.
Okay, very impressive. I think, it's an excellent approach. And quite frankly, as you see, you find that as you build data, you're going to develop more features that will be instantly available to other sectors other than what you're concentrating on, because that mobility is very important since you're mapping a large area, and it could change from point to point if you apply some monitoring technique to see how the water movement is affecting depositional structure. And then, it would then build on any improvement or degradation of that environment based on the data that you are currently measuring. But it's a very good approach. I think I'm quite impressed with the data and approach that you're using right now. I hate to bother you. I was involved deeply in reservoir simulation, and I see the challenge you have in dealing with some of this data. That's all I wanted to say. Thank you.
Yeah, really appreciate your feedback and engagement with that.
Okay, sure.
Do other people have thoughts, questions, suggestions? If not, been nice to spend some time with you. If anyone is interested in potentially providing some data, like I mentioned, the oxygen pH temperature from streams, rivers of any size that are of interest to you that are nearby. It's really just wide open if you want to run around and get some data, or if you already have some data for sites that are of interest. We can, as I said, run that through the artificial intelligence system and let you know what the priority scores are. If you want to, then we can send you a kit, get some additional data. If you know people that might want to do that, could be researchers, citizen science groups, classrooms, like whatever it might be, we'd super love to hear from, help us connect to other folks in those ways. Yep, and thank you, Bree. Put the Google form link into the chat. It'll just take probably a minute to fill out. And yeah, with that, it's thanks, Eric, for connecting us in here. Appreciate the opportunity.
My pleasure. Thank you for your whole team, everybody. Stefan, James, Brianna, Lupita, thank you for joining us today.